Hello guys, my name is Shmail. I will be teaching you and going over the whole biology specification, and I will at least attempt to, and so I hope whatever I'm teaching you is helpful. So let's begin with our first topic, cells. Right, there are two types of cells, prokaryotic cells and eukaryotic cells. Prokaryotic organisms have prokaryotic cells. And basically these prokaryotic cells are small and very simple. In comparison to eukaryotic cells, these are much more complex and much bigger. So you can look at the diagram here and see that prokaryotic cells have the size between 0.1 micrometers and 10 micrometers, whereas eukaryotic cells are between 10 and 100 micrometers, which is a big difference. And also you can see that the eukaryotic cells have much more organelles in comparison to the prokaryotic cells. So for today we will be focusing on eukaryotic cells, not prokaryotic, and in the next video we'll focus on prokaryotic. After that we'll focus on viruses. I haven't put viruses on this page due to viruses being a cellular, not being a cell. So let's look at eukaryotic cells. So eukaryotic cells, the examples of these would be animal cells, plant cells, fungal cells. These are all eukaryotic cells. So here is an animal cell. As you can see by the definition of eukaryotic complex, you can see that there are lots of organelles here, and so therefore it's complex. And so yeah, so don't worry about all these organelles labeled. I will go through each and every organelle in detail, so hopefully you will understand the function of these and the structure. So that's an animal cell, that's a plant cell, and that's an algal cell, and that's a fungal cell. So, cell membrane. So this is probably part of the structure of each and every cell. So every cell will have a cell membrane. So in the exam, do not, and I repeat, do not put the word cell membrane. That is GCSE level. We're not doing GCSEs. This is A level. So if you want to mention cell membrane, put cell surface membrane or plasma membrane, because you will not get the mark if you put just cell membrane. Well, it depends on what type of examiner you have, but no examiner is going to be lenient, so it's better off to put cell surface membrane. So the main job of this cell surface membrane is to control which materials pass in and out of the cell. So it's like a gate. It's like an electronic gate which controls what car goes in and what car goes out. So it's pretty much the same thing here. So basically this cell membrane is a double layer of phospholipids, also known as a bilayer of phospholipids, and they're arranged tail to tail with some protein molecules between. Now if you don't understand this right now, don't worry about it. In biochemistry you will go into this in much detail. This is just to explain what the membrane is made of. So right now I've just said that the membrane is made of phospholipids, and so the protein molecules are either embedded in the membrane or they penetrate the bilayer, forming pores. So there are pores in the membrane which allow large molecules, protein molecules, to enter. And these protein molecules are important in the transport of substances across the membrane. Proteins on the outside of the cell are often involved in cell recognition or as receptor sites for hormones. So the phospholipids are proteins and are not fixed in the position of the protein and can change. And this is called the fluid mosaic model, or the fluid mosaic structure. So if you don't understand this right now, all I want you to grasp from this slide is that do not call cell membranes, call them cell surface membranes, and they are made up of phospholipids and they control what goes in and out of the cell. That's all I want you to grasp from this for now. If you can grasp the rest bits, that's really good. So yeah. Nucleus. This is the largest cell of this is the largest organelle of the cell, and it's 10 micrometers long. Now, each and every eukaryotic cell will have a nucleus in the center or for plants, the nucleus will be in the corner. So every eukaryotic cell has this, except for red blood cells, RBCs. Now, you're probably thinking, why is a red blood cell classified as a eukaryotic cell? The reason for this is because, like I said in the previous slide, go back, that eukaryotic organisms have eukaryotic cells. Now, since red blood cells are found in eukaryotic organisms, therefore the red blood cell will be classified as a eukaryotic cell. And so if we go back, and despite it not having a nucleus, it is classified as a eukaryotic cell. So the function of the nucleus, it controls the cell activities. Now that's pretty basic, that is GCSE level, you should already know that. And so this nucleus contains a cell's DNA, which carries information that allows the cell to divide and carry out its cellular process. So when the cell is not dividing, the DNA is in the form of chromatin. So when the nucleus is about to divide, the DNA will also divide. But when it's not dividing, the DNA is in the form of chromatin. So as you can see in the bottom right hand corner in the diagram, in the micrograph, you have these little splotches in the nucleus. These little tiny dots are called chromatin. And the and this really dense dark patch you can see in the micrograph is called the nucleolus. And this produces the RNA needed to make ribosomes. So RNA stands for ribonucleic acid, and it's needed to make ribosomes. So the nucleus is bound by a nuclear membrane, also known as the nuclear envelope. And so this nuclear envelope allows nuclear pores, which allow things like messenger RNA to move in or out of the nucleus. Now, rough endoplasmic reticulum, or abbreviated as RER. Now, the endoplasmic reticulum joins to the nuclear membrane. So you'll find this surrounding the nucleus. As you can see in this micrograph, you have the nucleus labeled at the bottom, and you have the rough endoplasmic reticulum surrounding it. So in the exam, you might be asked to identify each organelle. If you are there trying to identify a rough endoplasmic reticulum, you know it will be next to the nucleus. It is a system with so sorry, that's meant to say thin, but it is a system with thin, intricate channels that spread throughout the cell. And where ribosomes, so basically on this rough endoplasmic reticulum, ribosomes attach to it. So you can see these little tiny dots on the rough endoplasmic reticulum, RER. These little dots are ribosomes. So the function of rough endoplasmic reticulum is that it folds and transports proteins made on the ribosomes. So folds and transports proteins. Now, cells which secrete and secrete large amounts of proteins will have large volume of rough endoplasmic reticulum because it folds and transports proteins, and so it will need to contain that protein. And so the rough endoplasmic reticulum also stores enzymes and other proteins. So small vesicles containing newly made proteins pinch off from the ends of the rough endoplasmic reticulum and either fuse with the Golgi complex or apparatus, or pass directly to the cell membrane. So we just had looked at rough endoplasmic reticulum. Now we're looking at smooth endoplasmic reticulum. Now, if we go back just one slide, the rough endoplasmic reticulum is called rough because it contains ribosomes on the outside. But smooth endoplasmic reticulum has no ribosomes attached to it, so it can't fold and process proteins. So, it occurs in small areas, not continuous with the nucleus, so it's not really next to the nucleus. Well, it might be, but usually it's not. And the little sacs at the end are called cisternae. And so you must know that word, keyword. And so let me just point out here what the cisternae looks like. So these little ends at the end of the SER, smooth endoplasmic reticulum, are called cisternae. So you might be asked to label a certain organelle. You never know. So just keep that in mind. So this is a site of steroids. It's where lipids is produced and processed. And this smooth endoplasmic reticulum also contains enzymes that detoxify a wide range of molecules and acts as a storage site. So what I want you to get from this is, that the smooth endoplasmic reticulum has no ribosomes attached, it's not near the nucleus, and the little sacs at the end of it is called cisternae, and it makes lipids. It produces and processes lipids. So, ribosomes. Now, I've already mentioned this before. It is on the rough endoplasmic reticulum. So, these ribosomes are small, dense organelles, around about 20 nanometers long. And most are attached to rough endoplasmic reticulum, but they can occur free in the cytoplasm as well. And there are two types of ribosomes, ATS and 7TS. ATS ribosomes are 25 nanometers in diameter and found in the eukaryotic cells, whereas 7TS ribosomes are 20 nanometers and found in prokaryotic cells. So if you're asked in the exam to tell them the difference between eukaryotic and prokaryotic cells, you can say that both of these have different sized or different types of ribosomes. And the function of these ribosomes, they carry out protein synthesis, as you know from GCSEs, and they assemble amino acids in the right order to produce new proteins. And so the ribosomes use the code on the messenger RNA to put amino acids together in chains to form specific proteins. So the ribosomes carry out protein synthesis, and proteins, as you know, are made up of amino acids. So the ribosomes assemble these amino acids in certain order to make new proteins, and then the ribosomes use the code on the messenger RNA to put amino acids together in chains to form specific proteins. And so general proteins that are to be used inside the cell are made by free ribosomes. And those proteins are to be secreted out of the cell, are made by the ribosomes on the rough endoplasmic reticulum. So, the Golgi apparatus, or the Golgi body, or the Golgi complex, I say Golgi apparatus, and Golgi vesicle and lysosome. So, also known as the Golgi complex, it is it's a tight packed group of flattened sacs, as you can see here. Now, please do not get confused between the smooth endoplasmic reticulum or the rough endoplasmic reticulum. This shape is quite distinct, as it will be like connected together. Let's move on and I'll tell you how you can identify it. So, it processes and packages proteins and lipids, and it's made in the rough endoplasmic reticulum. So this Golgi apparatus processes and packages lipids, which are made from the rough endoplasmic reticulum. It is also thought to synthesize or modify the chemicals before they are secreted from the cell. So these Golgi apparatuses, they they have these little sacs at the end, cisternae, and they just pinch off to make these things called Golgi vesicles, like here. And Golgi vesicles are constantly being added at one side and lost by the other. And these vesicles store proteins and lipids by the Golgi apparatus and transport them around the cell. So they're pretty much like a truck delivering food items. So this is the Golgi apparatus is a base, and then you've got a truck just delivering food to each and every store. And then you have lysosomes, and these are the small round organelles, and they are the type of Golgi vesicle. They're similar to Golgi vesicle, and they contain the enzyme lysozymes. And enzymes are kept separate from the cytoplasm by the membrane. And the function of the lysozyme lysozyme enzymes are used to digest invading cells or break down worn out components of the cell. Mitochondria is found in all living cells, living animal and plant cells, and is between 1 to 10 micrometers in length. And this is a site of aerobic respiration where energy is released in the form of ATP. This is to regulate metabolism, and they have a double membrane, and the outer membrane is smooth while the inner is folded, and the inner folds a large surface area, and this inner folded membrane is called a cristae. And they hold many of the enzymes concerned with respiration. And the central fluid filled space is called the matrix, and this is where enzymes are free roaming around, involved in respiration. So you know that any cell containing a huge amount of mitochondria, you know that that cell is involved or requires a lot of energy. So if we move on from mitochondria, the next one is chloroplasts. Now chloroplasts are found in plant cells, and the function of these chloroplasts is to make sugars by the process of photosynthesis. And chloroplasts have a double membrane. The first stage of photosynthesis is with light being absorbed by the chlorophyll, and the chlorophyll is within inside the chloroplast, and so the inner membrane is folded, creating thylakoids, while the membrane is made up of thylakoids, and this is in between this fluid called stroma. And in places the thylakoid membrane is arranged in stacks, and when the thylakoid is arranged in stacked up one on top of each other, it makes this thing called a grana. And these are thought to maximize absorption of light and transfer it into the chemical energy. And the stroma contains enzymes which fix CO2 into sugars, and adjacent grana are linked up by lamella. Now cell walls. Now cellulose cell walls are found in plant cells, and it consists of many cellulose fibers cemented together. So cellulose is a polysaccharide made of beta-glucose, and in the cell wall about 2,000 parallel cellulose molecules are packed together to form microfibrils. Now you'll learn more about these polysaccharide stuff in biochemistry, so if you don't understand it now, don't worry, you will find more about it later on in the videos. And so these like cellulose packed together bundle tighter to form fibrils, and this gives the cell wall great strength. And basically the function of this is to support and strengthen the cell to prevent them from changing shape. And bacterial cell walls are made from peptide doglycan, and fungal cell walls are made from chitin. Vacuole. These are found in plant cells only, and they could be found in animal cells, but majority of the time they're found in plant cells, and they're a cavity filled with a fluid called sap, and so it can take up 80% of the space in the plant cells, and this is the surrounding membrane is called the tonoplast, and the function of this vacuole is to absorb water by osmosis and swells, and this swelling keeps the cell rigid and stops the plant from wilting or becoming flaccid. And it also is a food storage, and so it stores things like sugars, and also it can help isolate unwanted chemicals in the cell. So that's it for eukaryotic cells. I've gone through each and every organelle and the structure and the function that you need to know for the exam. And so if there are any problems that you found with this video, do comment it down below, and if it was helpful, please do give it a like and subscribe. And yes, so in the next video we'll look at prokaryotic cells.